Genomic signal processing. Genomic signal processing is the field of analyzing and processing genomic and proteomic signals. It is a highly cross-disciplinary field involving engineering, biology, mathematics, and computer science, and has many important applications in both biology and medicine. Let's start with a bit of background information. A single strand of DNA is a biomolecule formed by a directional one-dimensional chain of smaller components called nucleotides. Each nucleotide has one of four possible bases, A, T, C, or G. A protein is another biomolecule that consists of many linked components called amino acids. There are 20 different types of amino acids in proteins, and like DNA strands, they are connected by chemical bonds into a long one-dimensional chain with a specific direction. The amino acid sequences in proteins are determined by the nucleotide sequences found in the DNA of the organism and are created in a process called protein synthesis. Each of the 64 possible triplets of nucleotides, known as codons, are mapped to one of the 20 possible types of amino acids, or a stop codon, which signals the end of an amino acid sequence. So you might be wondering how signal processing might relate to DNA and proteins. If the four nucleotides, A, T, C, and G, are assigned complex numbers, then a DNA sequence can be represented as a sequence of complex numbers which is essentially a discrete 2D signal. Triplets in sequences of nucleotides can be summed with weightings given to them based on their order to produce a complex number that can be mapped to the amino acid that they represent. For example, if we assign the nucleotide bases A, T, C, and G the values 1 plus J, 1 minus J, minus 1 minus J, and minus 1 plus J, the amino acid number can be calculated using the formula shown in Figure 3. For the ATG codon, Figure 4 shows that the amino acid value is 1.17 plus 0.88 J. The ATG codon would be mapped to the methionine codon on a complex plane, such as the mapping shown on Figure 5. Downsampling a sequence by a factor of n involves picking every n values from a sequence. Polyphase decomposition is when a sequence is expressed as the sum of m subsequences by downsampling by a factor of m. For example, if a two-band polyphase decomposition is applied to the sequences of consecutive positive integers, the polyphase components produced will be the sequence of odd numbers, H1, and the sequence of even numbers, H2. Protein synthesis can therefore be simulated by applying a low-pass filter followed by a three-band polyphase decomposition to collect the nucleotide triplets that make up the amino acids, and finally a vector quantization in which the three polyphase components are summed together after each being multiplied by some scalar values. Spectrograms are a powerful tool for analyzing audio signals to the point that trained people can know what word is being spoken in a voice recording just by looking at a spectrogram of it. One application of genomic signal processing is creating DNA spectrograms, a promising tool for DNA analysis. A DNA sequence can be represented by four binary sequences of the same length: an A sequence, a T sequence, a C sequence, and a G sequence. The nth element of the A sequence is 1 if the nth nucleotide in the DNA sequence has an A base and 0 otherwise. The same applies to the sequences T, C, and G, but with their respective base characters instead. This is a four-dimensional representation of the frequency spectrum of the DNA sequence, but it can be reduced to 3 dimensions, as only one of the nth values of the four numerical sequences can be 1, and the rest must be 0, so any sequence can be determined by the other 3 sequences. Imagine a regular tetrahedron centered at the origin, with each of the four bases assigned to one of the vertices of the tetrahedron. Each of the vertices is a distance of 1 from the origin. Three numerical sequences, XR, XG, and XB, can be defined using the A, T, C, and G sequences using coefficients taken from the tetrahedron's vertex coordinates. A discrete Fourier transformation would show that the frequency domain spectra of the A, T, C, and G sequences and the XR, XG, and XB sequences are the same, so no information is lost by this dimensionality reduction. To create a DNA spectrogram from the XR, XG, and XB sequences, discrete Fourier transformations are applied to a sliding window on each of these sequences to produce a frequency domain spectrum for each element in each sequence. This sliding window technique is known as a short-time Fourier transformation, and it provides local frequency information for all positions in the sequences. The magnitudes of the frequency domain spectra are mapped to the colors red, green, and blue, which are then superposed to produce a color in the spectrogram. In the DNA spectrogram shown, you might notice three high-intensity yellow regions at the frequency of 10. Properties and patterns in DNA sequences like these can be more easily noticed using a DNA spectrogram, which can lead to new discoveries about DNA structure. So what else is in genomic signal processing? Some other applications of genomic signal processing include gene prediction, which is the process of identifying which regions in DNA sequences encode genes and the functions of other regions of DNA. Gene regulation analysis, which is the analysis of regulatory DNA regions and proteins that control protein synthesis, and the diagnosis and treatment of genetic diseases such as cancer. Some other techniques used in genomic signal processing include deterministic models, such as Boolean networks and differential equation models, stochastic models like Bayesian networks and probabilistic Boolean networks, classifiers like neural networks and the k-nearest neighbors algorithm, and clustering algorithms like Euclidean distance clustering and hierarchical clustering.